Hello, everybody. I'd like to welcome everybody to the Development of Neurotoxicology talk and the seminar, the P30 seminar for this morning. I have the great pleasure of introducing an old friend from Vanderbilt, Dr. Mickey Aschner. He serves as the Harold and Muriel Block Chair in Molecular Pharmacology at the Albert Einstein College of Medicine. He has served on numerous toxicology panels, including the Institute of Medicine, the US EPA, the CDC, and the FDA. And he's a member of the Neurotoxicology and Alcohol Study section of the NIH. He serves on the Society for the SOT, Society of Toxicology, Council, and will assume the presidency of the SOT very soon. He has authored or co-authored greater than 640 peer-reviewed manuscripts and 100 book chapters. The research in his lab focuses on the following topics. One, modulation of C. elegans genes that are homologous to mammalian regulators of methylmercury uptake and cellular resistance, which then modify dopaminergic neurodegeneration in response to methylmercury exposure. Two, under a condition of methylmercury-induced oxidative stress, NRF2, he studies NRF2 that coordinates the upregulation of cytoprotective genes that combat methylmercury-induced oxidative stress. And he also looks at the genetic and biochemical changes that negatively impact NRF2 function to increase the vulnerability to neurotoxicity from methylmercury. Three, he looks at the PARC gene, which is a strong Parkinson's disease genetic risk factor, which can alter neuronal vulnerability to modifiers of cellular manganese status, particularly at the level of mitochondrial dysfunction and oxidative stress. His studies are designed to, one, shed novel mechanistic insight into metal-induced neurodegeneration, two, identify targets for genetic and pharmacologic modulation of neurodegenerative disorders, and three, to increase our overall knowledge of pathways involved in oxidative stress and develop and improve research models for human disease using knowledge in the environmental sciences. Mickey, it is so glad to have you here, and we look so forward to your talk. Thank you very much.
Thank you, Greg. Thank you, Greg. Thank you. Very nice introduction. I appreciate it. And thank you, Chris, for inviting me. This is wonderful because I haven't been able to do it in 2 years, to be honest. It's my first seminar. I've done many Zooms. I feel sorry for you guys at home, but it's obviously your choice. It's nice to see, though, life coming back to some normalcy. So it's a pleasure to be here. It's my first seminar in, I think, 2.5 years. So I'm delighted to be here. And first time in Louisville, too. I came in the afternoon yesterday. It's a beautiful city. And not to mention the weather. Okay. Let's get to science. So as Greg just mentioned, our lab is interested in gene by environment interaction. And the environment for us is basically metals. The endpoints that we are interested in are neurotoxic endpoints. I've been looking at mercury since 1980, and I picked up on manganese sometimes in the 1990s. So these are the two metals that we focus on the most. What I'll do today, there's not enough time to talk about everything, obviously, and I chose to talk about the manganese arm in our lab. I'll give you a brief introduction to manganese, and then we can ask basically the question as to what's the predictive value of doing studies in C. elegans. I had some of these discussions already today with some of John Wise's students. But hopefully I can convince you that we do see very similar things in C. elegans that we see in mammalian tissues, and that by doing studies in C. elegans, actually, we can predict mammalian neurotoxicity. So I've been studying for years, our lab has been studying for years, transport into different types of neurons. So I will discuss some transport systems that are important in terms of maintaining manganese homeostasis. And I'll talk about neurodegeneration, specifically as it relates to manganese. And the last question is some of the work that we're doing today. It will focus on actually MPTP, which is a compound that targets basal ganglia with a little bit of additional evidence to sort of close the cycle on my talk and manganese neurotoxicity. Okay, so why are we talking about manganese neurotoxicity? I think I also discussed it with somebody today. I was working on mercury for many years, and when I started to look at manganese, I realized that this is an essential metal, but we know very little about the physiology, specifically how manganese gets into the brain. And I thought it was important. I went to SOT the first time with my poster on manganese, and people were asking me, Mickey, why are you doing this work? You're crazy. And sort of I've been vindicated in some sense, because when I go to SOT now, there's about 50 posters on manganese and Parkinson's disease. So as I said to some of the students this morning, you know, if people tell you, why are you doing that? If you think it's important, keep working on it. So manganese is required. It's not like lead or mercury. None of us would be here if we didn't have manganese. It's ubiquitous. It's in every cell. And as listed here, I'm not going to read everything. It's important in many physiological processes. As far as the brain, it's very important in maintaining glutamine homeostasis, because the enzyme that converts glutamate to glutamine synthetase, which is localized to the astrocytes, is heavily dependent on the presence of manganese. And everybody's also familiar, I'm sure, with superoxide dismutase. It's the different types, but one of the isoforms is a manganese-containing enzyme, and it's very important in antioxidative stress. So, as you'd expect for an essential metal, we do have a daily intake. It's not very large, and we don't absorb very much because we are at a steady state. So we basically make up for what's lost. So generally, absorption of manganese is in the range of 3% in adults. It's a little bit higher in neonates, young children. I won't talk about this, but we've actually done some studies in neonates that are being fed by parental nutrition, looking to see whether they have too much of an MRI signal for manganese in the brain, because manganese is a paramagnetic metal. But I won't talk about it today. But there's a translational value to looking at manganese in neonates. Once it's absorbed from the gut, it goes through the liver. Most of the manganese is in the liver, and then it circulates in the blood. It binds to different globulins, and then it's taken up by different tissues. Now, as far as the neurotoxicity of manganese, we're focused on this topic because exposure to high levels of manganese have been associated with a Parkinsonian-like symptom. For those of you that are interested in neuroscience, manganism, which is the term that's been coined for overexposure to manganese, is not identical to idiopathic Parkinson's disease by any means, but there are a lot of similarities. There are very distinct features to each of them, but you get dopaminergic neurodegeneration in both of these disorders. So it is it's a Parkinsonism-like disease. And I'll talk about it a little bit more. But basically, the reason probably that manganese accumulates in the same area that you find iron, as indicated in this slide, in the basal ganglia, which are areas that are important in terms of motor function, caudate putamen, globus pallidus, subthalamic nucleus, and so forth, is that manganese tends to share transporters that are also transporting other divalent metals or trivalent metals. So I'll get to it in some detail. Something like the divalent metal transporter 1, for example, is sort of a very promiscuous kind of transporter. It likes zinc, it likes manganese, cadmium, arsenic, and many other metals. So when you have too much manganese on board, what you might do is inhibit the transport of iron, and vice versa. If somebody is iron deficient, you might find that they have higher burdens of manganese in different tissues. So if there's one thing to remember from the seminar is that there's competition between these types of metals. Now, there are a number of populations that are at risk. I mentioned briefly parental nutrition. We have done some calculations, and again, I won't show you any of the data. Somebody who receives parenteral nutrition probably receives about 100 times more manganese than they would receive if it's a neonate from breastfeeding from their mother. So there's a tremendous amount of manganese in parental nutrition, and I think there's really no reason for it. It can be reduced. We have done some MRIs on these kids, and if you do it, it's been published a few years ago, you will see a very typical T1 weighted image indicative of accumulation of manganese in the basal ganglia. So parental nutrition is one. Iron deficiency, I mentioned already, because of the nature of the transporters. Individuals that are iron deficient, and that's very prevalent, as everybody here knows, in old people, in pregnant women, poor diets. So there are billions of people actually around the globe that are iron deficient. Iron deficiency will upregulate the iron transporters. If there's no iron around or reduced level of iron, the DMT1, the other transporters, would take up other divalent metals, one of them being manganese. Liver disease is another one because manganese is predominantly secreted in the bile. So people that have liver disease develop what's known as hepatic encephalopathy. That will lead to increased levels of ammonia, for example, in the brain, increased levels of manganese as well. And then there's some environmental exposures. I'm not going to say very much about them, but high levels of manganese in well water. It's not similar to lead, but there are many homes, especially in rural areas, that are not on city water supplies. It depends on where you live around the US and other countries because manganese is in the soil. It has been shown that levels of manganese that exceed 300 micrograms per liter can lead to neurobehavioral deficits in children in terms of various kinds of behaviors. MMT is sort of not an issue in the United States, but it is an issue around the globe. It's an oxide of manganese, and we seem to have been we seem to have been poor students of our history, as is manifested these days in Europe, but also in terms of environmental issues. We took out lead, and what are we doing now? We're putting other anti-knock agents in gasoline. Not in the US again, because the EPA has been fighting the company that makes MMT, so it's not being used here. But it's used to the north in Canada, it's used to the south in Mexico, it's used in China, it's used in many countries because it's very cheap. It's an anti-knock agent. Basically what it does, it makes the engine work better, more efficiently. And it has been shown actually that in places such as Canada, atmospheric levels of manganese have increased by about twofold over the last 2 decades. So they're very low. And the question is, what does it mean? The answer, I think, is fair to say we don't know. But you can imagine somebody exposed to twice the amount of manganese that was in their breathing air for their lifespan. And it's probably a question that has some scientific relevance, health relevance. Welding is another issue. It's complicated because when you weld, the fumes are not just manganese, there's a lot of iron. But the rods that are being used for soldering are basically 100% manganese. So if you work, even if you put your mask on and you inhale, you breathe normally, there's a lot of manganese that can be absorbed. And there are studies actually from WashU, from Dr. Brett Rosette and his group, showing that welders develop Parkinson's disease or Parkinsonism-like disease at a much younger age, about 7 years younger than the population at large. So it's about 55, 56 years versus a mean of about 63 in the general population. And the last one is related to drug abuse, methcathinone. It's a product of pseudoephedrine and ephedrine. You won't find them anymore on the shelves in CVS and other places because you can oxidize those compounds with potassium permanganate. Permanganate is manganese. For those of you that know the MPTP story, it's very similar. It's okay now. Okay, thank you. It's very similar to the MPTP story. You don't tend to see people in their teens or 20s coming into the emergency room with Parkinson-like syndrome. But if you consume methcathinone at high levels, you may end up with symptoms analogous to Parkinson's disease at a very young age. So there's been sort of not a pandemic, but it's more endemic. It started in Eastern Europe and it's moved through Europe. And there are some papers about it for anybody interested, including New England Journal of Medicine. So, from the toxicology point of view, again, the brain is probably the most important target organ. And I have this just to show you that you don't have to have the highest concentration of anything in the brain in order to induce neurotoxicity. You will see here in the slide that the level of manganese in the brain in humans, rats, rabbits, all kinds of different species are not necessarily the highest, but the target organ for manganese is the brain. So there's something special about the brain that renders it more sensitive to manganese versus liver or other tissues. And sorry, the neurological consequences of manganese exposure are, as I said before, very similar to Parkinson's disease. In early stages, there's the psychiatric or psychological disturbances, madness, sort of locura means craziness. So locura manganica refers to manganese. And then you develop an extrapyramidal syndrome, which is very similar to what you see in Parkinson's disease. The reason for that, I don't have a slide here, is if you look at the pathways that connect the substantia nigra and the globus pallidus and other areas, the one that's affected, the area of the brain that's most affected in Parkinson's disease is the substantia nigra. That's where the dopaminergic neurons are. So when those neurodegenerate, you develop the kinesias, all the characteristic symptoms of Parkinson's disease. With manganese, that's the area that's being affected is an area that actually gets projections from the substantia nigra. It's known as the globus pallidus. There's an internal, external capsule, the globus pallidus. So when I try to explain it, I basically say, think about this pathway as being a hose. It's much more complicated than those two brain regions, those two nuclei. But it really doesn't matter if you tighten the hose at the origin or at the end. The water is not going to flow out of the hose, whether you restrict the flow in the beginning of the hose or at the end part of the hose. So the symptoms are very much the same, although, as I said before, there are some distinct differences. Okay, so I'm going to move now and talk to you about some of the transport studies that we have done. Are there any questions on this part? Is everybody okay? Okay. Okay, so I show this slide. It's not even contemporary. It's probably outdated by a couple of years by now. We're talking about one metal, and there's probably about 14 or 16 different transporters for one metal. So it's obviously very complex. And I've been asked, why do we need so many transporters for one metal? One would think that having 2 or 3 would suffice to sort of make sure the homeostasis of manganese is 100%. It can be maintained at whatever level is necessary. Levels of manganese in cells are in the range of 75, 100 micromolar. So it's pretty abundant. But as you can see, there's many, many transporters. And I don't have a good answer as to why we have so many. It may be that the concentration of manganese might have to be regulated at different levels in different types of organelles. So we know very little actually about the distribution of the different transporters within the cell. But the bottom line, we have many of them. And I don't know if you can see can you see yeah, I don't know if I can do it, but anyway, I'll try to point to some of them. It's too hard to do, but on the top on the left, you can see DMT1. I mentioned it. It's in the sort of blue color. I'll talk about this one a little bit. You can see underneath in purple is a calcium manganese a channel. If we go down, there's a ZIP8, which again transports manganese and a number of other metals and cations. The one at the bottom right, if you go back to about 7 o'clock in green, that's actually an efflux mechanism. We discovered a few years ago that ferroportin is one of the means by which manganese can be actually extruded from the cell. So again, here you have an iron connection because ferroportin is also the main transporter for iron, exporter. And if we go down to the right to about 4 o'clock, you'll see SLC3810. And that's the one that I'll spend most of the time on today, which is the transporter that we discovered a few years ago, again, being an exporter of manganese. So we know of probably about 10, 12 importers for manganese and 2. That does not mean that there aren't many more. It just means that we don't know what they are. Okay, so here are some studies that we did with C. elegans, and I apologize, I'm going to show these slides before I tell you about the model. So I will show you a couple slides later about the advantages of using C. elegans. But basically, these are some studies that we looked at trying to determine whether DMT1 is a transporter for manganese in C. elegans. We knew it already actually from mammalian studies. So here it's more, is it predictive? Is this model predictive of mammalian transport of manganese? And what you see here is the sequence homology of the manganese transporter DMT1. In each panel, going from left to right, there's from the bottom, it goes up from east all the way through C. elegans and then to I believe humans. I can't see it from here. But the point of this slide is to show that there's a great homology between mammalian and all the way to east. Everything which is in the light blue color is homologous between all these species. So there's probably about 70% homology in DMT1. That's important because if you're studying a transporter, you want to make sure it has homology to the mammalian system. Now, the nomenclature is a little bit different. Unfortunately, DMT1 is referred to in C. elegans as SMF, and there's 3 different isoforms, SMF1, SMF2, and SMF3. So the first question that we had was very simple. If we delete SMF1, SMF1, SMF2, or SMF3, what's going to happen? And the hypothesis was if you delete them, the animals should be more protected, right? They will take up less manganese. There might be other transporters that may compensate for it, but we wouldn't have expected to see more toxicity. The toxicity endpoint in this case was survival. So it's a very crude measure. So on the y-axis is survival, percent control, and on the x-axis is a log scale of the concentrations of manganese. And there are 4 different strains here. The wild type are in black, and then we have SMF1, which is SMF1, which is blue, SMF3, which is the maroon color, and SMF2, which is in green. So what would you expect? If you would expect that if you knock out SMF, any of the DMT1s, that the animals should be less sensitive. Less sensitive means that you would expect to see a rightwardly shift in the LD50, right? So that happens with actually two of the strains. SMF1 and 3, you can see the curve moves a little bit to the right. I've listed the LC50s, the lethal concentrations 50s. You can see that for SMF1, it goes to 94 millimolar. It doubles compared to 47 in the wild type, and it goes up to 128 in the SMF3. So if you knock out those two isoforms, they seem to be more protected. But what was actually very surprising to us is that the animals where we knocked out SMF2 became more sensitive to manganese. So that necessitated some additional studies, and this was actually still done at Vanderbilt. I had a very good postdoc at the time and a great graduate student, Alexander Benedetto and Catherine Au, and they've used some ICP-MS studies, and then they did some expression patterns looking at transcriptional and translational GFP fusions. And what we found was that indeed the SMF1 and SMF3, as shown here, have the SMF2 have significantly higher concentrations of manganese compared to the wild type animals, which was something that we did not expect. So I'm going to show you a couple slides with different expression patterns for SMF1, 2, and 3. Again, this was done by transcriptional and translational GFP. At the bottom of the slide, you see basically the whole worm, schematic of a worm. To your left is the head, to your right is the tail. And the GFP is in the green, in the upper panels. And if you look at the summary, the schematic at the bottom, what you'll see is that SMF1 is predominantly in the gut and sort of under the skin, under the cuticle of the worm. And there's a little bit in the head region, again to the left in the pharynx. And if you compare it to SMF2, it's very different. Now we're looking only at the head region. So we're looking at the sort of the 1/3 of this worm on the left side. Okay, there's no expression of SMF2, which is sort of the outlier between the three, the one where the animals actually had the higher concentrations of manganese and they were more sensitive to manganese. In this case, all of the SMF2 is expressed in the head region, in the area of the pharynx. And if you look at SMF3, which was the LC50 was going in the same direction as the LC50 for SMF1, you find pretty much the same kind of distribution. Okay, so it's all over the place, mostly under the skin, the cuticle, and in the gut area. So we've done a number of additional studies on this. Basically, this is a summary. It's a working model that we developed, which basically states that SMF1 and SMF3 are the real sort of isoforms of DMT1 in the worm, and they are responsible for the uptake of manganese, whereas SMF2, again, the worms were more sensitive to this deletion, seem to be more of a, SMF2 seems to be more of a sensing and an isoform that regulates the pumping of the pharynx to inhibit the export of manganese from, or the uptake of manganese from these worms. The next transporter that I want to talk about is SLC3810. So this is now an exporter, not a transporter. And this got our attention, I think it's about 8-10 years ago now. We came across a paper that was published actually on a human population, a family in Italy, that had shown that people that have mutation in SLC3810 develop symptoms that are very similar to Parkinson's disease, and it was characterized by elevated levels of manganese in their blood. They had what's referred to as hypermanganesemia. So very similar. They actually characterized two of the mutations. You can see them, a missense and a nonsense. And you see the structure. It's indicated by the red star, and there's a yellow dot there. for those two different mutations. So what we've done with a colleague at the University of Texas was develop basically an hypothesis that given the clinical picture in these patients, this may be something that's involved in the transport. This transporter may be involved in maintaining levels of manganese in cells. And the hypothesis was that SLC3810 would facilitate manganese efflux. And in this case, we used HeLa cells. So it would protect HeLa cells from manganese toxicity. If we talk specifically about the expression of SLC30 in dopaminergic neurons, which are sensitive to manganese, the hypothesis would be that if you have mutation in SLC3810, this would result in elevated levels of manganese in regions where dopamine is present, in basal ganglia, basically. And animals with these mutations would be more sensitive to manganese toxicity. So the first thing that we did was to try and localize this transporter in different cells. On the left top panel, what you see in green is the expression, or going from the top, all the green is basically SLC3810. The red, the middle panel is connexin, which is just a marker for the endoplasmic reticulum. And on the right, you have the merge of the two panels to its left. So the first thing that you can appreciate is in the wild type, if you go all the way to the top, the wild type SLC30 is almost all in the cytoplasmic membrane, okay? Now the five images underneath the top one are different types of mutations in SLC3810. Yeah, there's no pointer, right? You have a pointer here? Where is it?
Oh, I'm sorry, your mouse was pointing.
Okay, it's okay. If I go away, they won't hear me. So, okay. So the rings are the SLC3810, and if you go down the five panels in green, these are different mutations. I think you can appreciate that the distribution of SLC3810, once you mutate this gene, are very different. It's not in the membrane. It's very diffuse. The connexin is pretty much the same. It just labels the endoplasmic reticulum, so we don't have to talk about it. And you can see in the merge, SLC3810, again, with the red, it's all in the membrane, and you get an orange color with the mutations because the SLC3810 overlaps basically with the marker for the endoplasmic reticulum. It doesn't mean it's binding to the endoplasmic reticulum, but it means it's not in the membrane. So you can see very distinct differences. Actually, if you go to the top, and this is the quantitative measurement of different panels and repeats of what is shown in the fluorescence studies on the left, you will see that 100%, nearly 100% of the transporter in its wild type is in the membrane, and you can barely find any transporter once it's mutated in the membrane. We've also done some bioaccumulation studies that basically tells you if something is in the membrane or it's not. So if you look at the Western blot underneath, you see one protein, which is GPP 130. I'll talk about it in just 1 minute, but GPP 130 is not a membranous protein, so you don't see any band. And if you look at the wild type SLC3810, you can see a band. So again, indicative of the fact that it's in the membrane. And if you do the same kind of blot with some of the mutated forms of SLC3810, again, you don't see any band because the bioaccumulation study, as I said, indicates whether something is membrane bound. So you don't expect, based on the fluorescence, to see anything in the membrane. So the first thing that we learned from this is that the SLC3810, the mutated forms, are not in the membrane, which would suggest that they're not functional, right? They're not in the right place. Okay, I know these are a little bit complex, and it's even more complex when you don't have a pointer, but please bear with me. Okay, so what are we doing here? So here we're looking at the protein that's basically responsive to manganese, GPP-130. GPP-130 is a chaperone of manganese. So what it does, it takes the manganese from endoplasmic reticulum and it transports it to endosomes and lysosomes. So as the concentration of manganese in the cell is increased, the GPP translocate the manganese and the GPP itself gets degraded in the lysosomes. So let's go back to the top left again. You see there's, it's mostly red. That red thing is GPP-130, okay? So these are wild type. Well, this is just the controls. GPP is fluorescent in red. We move a little bit to the right. There's another GPP-130, and there's a bar on the top that says manganese. So when you treat the cells with manganese, the red dot disappears altogether, okay? Can you see that? Why is that? Because the GPP, there's a lot of manganese in the cell. The GPP moves the manganese to the endosomes and lysosomes. The GPP gets degraded, so there's no more red fluorescence, okay? The panel underneath, we start with a red dot, okay? And you can see that there's a ring, because what we've done here, we've transfected the HeLa cells with SLC30A10, okay? Now we move to the right, we treat with manganese, and the red dot is preserved. Can you see that? Why is it preserved? Because now we have a complement of SLC30A10 in the cell, which is able to export the manganese from the cell. So you don't need essentially the GPP-130 to move the manganese from the endoplasmic reticulum to the endosomes and the lysosomes. It's the SLC30A10 that gets rid of the intracellular manganese, okay? And the GPP is therefore preserved. And on the right, the panel is going all the way down. You see basically the same thing. You see that for the wild type, when you treat with high levels of manganese, the red dot is preserved. Okay, when SLC30A10 is there, because you don't need the GPP-130 to move the manganese to the endosomes and lysosomes. You can get rid of the manganese by means of the SLC30A10. And the 5 different treatments underneath is with the different mutants. You can see that in all these conditions, when you treat the manganese with GPP-130 disappears, because you don't have the means to get out the manganese, okay? So again, this is another indication that the SLC30A10, the wild type, is functional. It's able to reduce the concentrations of manganese in the cell. And we've actually tested this directly. So this is very simple. On the left, you have manganese concentrations. The black bars are the controls, and the gray bars are cells that express the wild type SLC 30A10, and you can see that if you measure the concentrations of manganese upon the different treatments, in each case you'll find higher concentrations of manganese in the controls, those that do not have the complement of SLC30A10s expressed in them. We did some chase experiments, and that's again very consistent with SLC30A10 being an efficient exporter for manganese. If you look at the controls, again in black, and the SLC30A10, the wild type, you see that in these chase experiments, the levels of manganese in the gray bar in the transfected cells are lower than the controls. There's more actually on the outside, more manganese. And if you look all the way to the right, in one of the mutants, there's no difference. No difference because there's an expression of a transporter, but it doesn't dock in the membrane, and it's not functional. And finally, almost finally, for this part, SLC protects cells against manganese toxicity. So this is very simple. We're looking at relative cell viability with different concentrations of manganese. The control cells, as you expect, they're dying. Can you hear me? I think we lost it again. I think it's okay now. Okay, so the control cells are dying as you increase the manganese concentration, as you'd expect, but the death rate, the survival is higher in the cells that have been transfected with the wild type SLC. Okay, so they're protected. And when we first sent this for a review, you know, everybody was asking for the relevance of this, so we had to add some additional studies in mammalian, well, HeLa cells are mammalian, but in cells, neuronal cells. So this was done in midbrain cells that express dopamine, which is, again, the relevant neurotransmitter for manganese. And we've looked at all kinds of different parameters, the longest primary branch, the number of branches, the arborization of the different dendrites and so forth. You can see that the results are basically the same. When, again, if we look just at one of those, let's look at the one on the top left. We're looking at longest primary neurite per neuron, okay? So we have 4 different groups. We have the controls, and we have the mutants in black without manganese. And then we have the same neurons that have been that express the wild type SLC3810, and to the right, cells that express one of the mutant SLC3810. And you can see that when you treat with high levels of manganese, the gray bars, the cells that express SLC3810 look exactly like the controls, okay? Again, indicative of the fact that they can most likely export the manganese from the cell, so you don't see any toxicity, whereas the mutants are very sensitive. 50, 60, 80% reduction in all these parameters. Now we've taken that to C. elegans as well, because we wanted to do some studies in vivo. This is in the botanical gardens, actually, in New York. And I'll tell you a little bit about at least in my opinion, why it's a very predictive and useful model to use C. elegans. So they have egg to egg is a very short period of time. It's about 50 hours. They are very easy to feed. They eat E. coli. You can freeze them. It's relatively easy to generate all kinds of different strains. You can freeze them. If you're interested in neurotoxicology, they have 302 neurons. It's an advantage, but it's a disadvantage. They have 8 dopaminergic neurons. So again, we have millions. I don't know how many millions actually, but it's a very simplified system. Surprisingly, they actually have a lot of genes. They have 19,000 genes. And there's an RNAi library, I talked about it with some of the students, that has 16,000 of those genes. What does that mean? Since they eat bacteria, what you can do is you can give the RNAi of interest to the bacteria. You give the bacteria to the worms, and the next day you have knocked down or deleted or mutated a given gene. So you can ask questions about what's the function of a gene that's there and present. Vice versa, you can take genes that are not expressed in C. elegans. You can take alpha-synuclein, for example. If you're interested in Parkinson's disease, you can express it. And you can ask simple questions about what's the function of alpha-synuclein. Is the wild-type gene neuroprotective? Is the mutated form the one that's responsible? Or which one of those is the most responsible for PD? This is not my boat, but somebody actually sent it to me. I've never seen this boat, but I thought it was cute. Okay, so what we decided to do was, first we decided to see whether SLC3810 is expressed in worms. And if you remember, when I talked about the DMT1, I mentioned you want to see high homology, sequence homology between mammalian genes and other species, if they're non-mammalian, invertebrates. So in this case, we actually couldn't find anything that was of high homology that resembled the SLC3810. The highest hit was like 38%. We didn't know the function of the gene. So what we decided to do is to get human DNA and then transfect the worms and express the different mutant forms of SLC3810 in the worms. And I don't know if this will work. Before I show you those data, so Okay, yeah, okay. So this is one example of a worm. It's about 1 millimeter in size. And you can see there's some dots, fluorescing dots in the head. Can you see them? And there's a couple of them in the tail. So now the lights are off. You can see 6 dots moving in the head region and 6 dots moving in the tail region. So these are the 8 dopaminergic neurons. What's been done here is to tag the promoter for the dopamine transporter, DAT1, with green fluorescent protein. So the green fluorescent protein is only expressed in cells that have the dopamine transporter. And there's 8 of them. I'll show you, you can do it with all kinds of other neurotransmitters. So you can ask some specific questions about the sensitivity of different neuronal phenotype to manganese in this case, but you can obviously use chromium or whatever you're interested in. So this is what you see with manganese. So there's 4 different, if you go from left to right, there's a PDAT, 1-GFP. So this is a green fluorescent protein of the dopamine transporter. The one to the right is the PUNC47-GFP. This is a specific label for GABAergic neurons, PUNC17 cholinergic neurons, and then there's one in red, which is DIL, a chemosensory neuron. And the top is 0 millimolar manganese, and if we go down, it's 50 millimolar manganese chloride. And we have to use, I should actually mention this, we have to use relatively high concentrations of manganese and everything else because the worms have a very thick cuticle. So, but if you measure, and I showed you one, I should have mentioned it there, if you measure the concentrations of manganese in the worms themselves, they're very similar, even if at these concentrations, to what you find in mammalian systems in the brain. Actually, we published a paper on it. We did some calculations. It's exactly the same. So these are not irrelevant concentrations. It's just we have to treat them with very high doses of manganese because not very much gets in. But in this case, what you can see is intact, different phenotypes on the top. And when you add the manganese, you can see that the only neurons actually that degenerate are the dopaminergic neurons. And this is, again, very consistent with what you see with mammalian systems. GABAergic seem to be spared. The DIL, the cholinergic neurons, seem to be spared. Now, the nice thing about C. elegans is also that you can do functional studies. So you can couple the morphological changes that I've shown you in the previous slide with a behavioral test. In the worm, the dopamine drives food-seeking behaviors, okay? So the worms are very much like we are. If you go to McDonald's, usually you get your burger, right? And you sit down for a few minutes. You slow down and eat. If you present the worms with bacteria, they don't stop moving, but they slow down. And there's a delta to it. It's about, they move, the number of body bends is about 1 per second. So if you measure 20 or 30 seconds, it will be 20, 30 body bends. If you present them with food, the body bends would go from, let's say, 20 per 20 seconds, they'll go down to 10, okay? 12. So the delta is about 5 to 8. So we can measure the delta, the change in the presence and absence of food, okay? And that's what we've done here. So if we focus on the different bars, what we have here is 3 different strains. We have the controls. We have worms that express the wild type SLC3810. And then to the right, sort of the graded grayish color, are worms that express a mutant SLC3810. So you can see worms with wild type dopaminergic neurodegeneration, they're all at 100% in the absence of manganese, 0 manganese. If you treat them with 10 millimolars of manganese, okay, now you could see that there's about 50% neurodegeneration in the animals that are wild type. No expression of SLC3810, either wild type transporter or mutant transporter. The animals that have been exposed or transfected, or they express SLC3810 wild type, are doing a little bit better. Okay, the neurodegeneration is slightly lower. You preserve about 70% of the dopaminergic neurons compared to 50% when you don't express the SLC38 wild type. And in the animals that express the SLC3810, the mutant form, they're just as bad as the wild type, if not worse, when you treat them with manganese. Underneath is the change in the number of body bends, the behavior that I explained. So again, the three groups, the three different strains in the absence of manganese, they look the same. If you look at the three different strains now in terms of the number of body bends in the presence of manganese, you can see corresponding to the change in dopaminergic neurodegeneration, you see that the animals are not able to slow down, okay? The delta is smaller. What does it mean? That they're not able to slow down. In the absence, in the wild types, they're not able to slow down. The delta is smaller, and the delta is much larger now. It's almost the same. Goes back to almost 8 when we express the SLC3810. So having SLC3810 reduces the neurodegeneration and also enables them to slow down when you present them with food. Okay? So these two are, or at least the last one, the body bends, is a functional correlate of manganese toxicity. So to summarize this, and then I have 5 more minutes if it's okay. We think SLC3810 is an exporter of manganese. You can see it's in green, and all of it is in the membrane. It's functional in terms of being able to dock to the membrane and then export the manganese. And the mutated forms, we've worked with 5 of them, including those 2 that have been identified in the human population. We don't know exactly where it is, but it seems to be, well, it doesn't seem to be in the cytoplasmic membrane. So it's dysfunctional, and we think that's the reason for the increase in manganese concentrations in these individuals. And the last thing I want to touch upon is the role of dopamine in neurodegeneration. So these are actually ongoing studies, and I'll show you what we're doing. So again, those of you in neuroscience would recognize that the synthetic pathway synthesis and catabolism of dopamine and serotonin are very similar. If you treat worms with manganese and you look at wild-type serotonergic neurons, you won't see anything. And that's why we chose serotonergic neurons. What we decided to do was to re-engineer the serotonergic neurons to make dopamine. And it's not actually very hard to do in C. elegans. So you see the pathway to dopamine synthesis. You can see that there's only 2 differences. There's the CAT2, which is tyrosine hydroxylase in mammalian systems, which is the rate-limiting enzyme in the production of dopamine. And if you go down, you see DAT1, which is a specific transporter for dopamine. Okay, so these 2 are inherent to dopaminergic neurons. And if you look at serotonin, you see TPH1, or tryptophan hydroxylase, is an enzyme that's responsible for serotonin synthesis. And the reuptake of serotonin is by means of by serotonin reuptake systems, by SERTs. So MOD5 would be one of the transporters. So what we decided to do is knock out TPH1 and MOD5 in the serotonergic neurons and express CAT2, or tyrosine hydroxylase, and DAT1 in the same neurons. So at this point, if we are successful, they should not be making serotonin, they should be making dopamine. And then the question becomes, are they sensitive to dopaminergic neurotoxins? So this is the way it looks. Again, I won't belabor the methodology, but these are now ADF neurons. They are serotonergic neurons, and because we express DAT1, that's with a promoter, GFP promoter, hooked up to DAT1, you can see that the DAT1 is expressed in the serotonergic neurons. And if you look at these neurons, this is what you'll see. So the left 3 panels going from the top to the right These are CYP and ADE neurons. These are just normal dopaminergic neurons, okay, in green, DAT1. The blue in the middle is a formaldehyde-induced fluorescence, which can detect dopaminergic neurons. So you can see if you merge the two at the bottom, in wild type worms, the dopamine is expressed the dopamine is expressed in cells that have the DAT1, which is nothing new. In the middle, the green is N2s, which are wild types. Again, you can see the fluorescence. You can see the fluorescence upon manganese exposure. It goes away. You lose some of the dopamine fluorescence. And if you go further down, these are animals that are knockdowns for CAT2, so they don't have the ability to make dopamine. Again, you don't see any, as you'd expect, the dopamine fluorescence. And to the right, you see these new worms, MAB371. These are the ADF neurons. This is very similar to what I've shown you in the previous slide. These are serotonergic neurons that make dopamine. And if you look for dopamine, you can find it by using the formaldehyde-induced fluorescence. And you can see the dopamine overlaps with the expression of PDAT. So now we have serotonergic neurons that make dopamine, and the question becomes, when you treat them with something like MPTP or manganese, are they going to be sensitive to manganese or MPTP? So I'm going to show you MPTP because so far the data is cleaner with MPTP. What you see on the left is the ADF neurons. These are fluorescing for PDAT, for the dopaminergic transporter. In the qualitative or quantitative data that you see with MPTP, on the y-axis on the top, you see worms with wild-type ADF neurons. So we haven't touched them. They express serotonin. When you look at these neurons, they do not degenerate at different levels of MPTP. And if you look at the panel below, you look at neurodegeneration in worms that have been re-engineered, where the serotonergic neurons are now making dopamine. And you can see that with high levels of MPTP, there's about 80% neurodegeneration. So it seems that dopamine is required for the neurodegeneration of these cells. Now we've done something very similar with manganese. We've asked the same question, is dopamine necessary? We're still doing some studies, as in the previous slide, we haven't finished them. But another approach was basically to say, well, if dopamine is necessary for the neurodegeneration, then if we knock out PDAT, which is the transporter for manganese, and if we knock out tyrosine hydroxylase, or CAT2, which is the rate limiting enzyme for the synthesis of dopamine, and these cells don't make, or the worm doesn't make dopamine anymore, then these cells should be preserved. We should not see any neurodegeneration. So that's what we've done here. There's 4 different panels. There's the wild type, CAT2 are, there's no tyrosine hydroxylase, so these worms don't make dopamine. DAT1 is the transporter for dopamine, okay, not present. And then we made a double mutant, no CAT2 and no DAT1. And the 4 panels on the left with 0, you can see there's no differences in terms of dopaminergic neurodegeneration. This is still with MPTP. You move to the right, you can see that you get about 50% neurodegeneration in the wild type. Okay, so these have dopamine, you get 50% neurodegeneration. If you knock out CAT2, DAT1, or you make a double mutant, you don't see any neurodegeneration. Again, very consistent with the previous slide, meaning basically that you need dopamine for the neurodegeneration. And this is the manganese, actually. As I said, it's not as clean. We have to repeat this and probably add some more animals, but the trend is definitely there. The CAT2, you see maybe 40% neurodegeneration, and if the worms or these neurons don't make the dopamine or don't have the ability to reuptake the dopamine, or both of these systems are knocked down, you can see they seem to be more resistant. Again, consistent with the fact that dopamine is necessary for the neurodegeneration. And I'll leave this slide on. So hopefully I've shown you that dopaminergic neurons are sensitive to manganese. Hopefully I've shown you that there's some predictive value to use C. elegans. We can couple morphology, we can couple functional assays to look at the relationship between different metals, in this case specifically the manganese and neurodegeneration. I think it's a great tool because, as I've shown you in the last part of the slide, you can actually re-engineer neurons to make neurotransmitters other than the ones that they are designed to express. And these are the people that have worked on this. Pen Chan has been in my lab, actually came from Vanderbilt. Julia Bornhorst, mentioned it to you. Get in touch with her. She does a lot of analysis, and I think she works with chromium. Mahfouz is finishing his Ph.D. He did the work in the last part. Some of the work on the HeLa cells was done with Shamshuvra down at the University of Texas, and my collaborator for many, many years, Aaron Bowman, and last but not least, this actually all started with a Gerber Foundation grant to look at neonates that are exposed to manganese in parental nutrition, which I didn't talk about today, and one of these grants is actually on mercury. So, and I like this slide too. So, truth passes through 3 stages. First, it's ridiculed. Second, it's violently opposed. Third, it's accepted as self-evident. So, hopefully, we're between the second and third stage. Thank you. Thanks again for inviting me. Chris.
Great talk. Really fascinating. So, do worms have monoamine oxidase?
Yes, they do.
So, it may be following the same sort of metabolic activation that ordered the improvements?
Yes, they do.
It requires the dopamine, which is why it targets.
Yes, yeah, that's correct. They also have actually astrocytes. We talked about this morning. So pretty much what you see in terms of the astrocytes is that like each of the cells in the worm is paralleled by an astrocyte and a projection. So it's not, there's more astrocytes actually in human brain than neurons. But it seems to be a 1-to-1 ratio in C. elegans. But they do have monoamine oxidase, so in terms of the MPTP, I would expect that it's the same kind of pathway. You get MPP plus generated, yes. We haven't done anything with MPP plus by itself. It's more polar, so it's probably you'd need even higher concentrations. But yeah, it should readily get in and pretty much the same pathway. Yeah. Johnny, let me go with Johnny then.
So I was struck by your HeLa cell work, with manganese transporters going down after manganese exposure. I'm wondering, do you see that in human populations that are exposed to manganese? It's like SLC and GP.
So I mentioned that, there have been different studies now, Quadri, there have been different groups actually that publish similar results in human populations. There are some studies on SNPs in DMT1 in relationships to Parkinson's disease. There's a study, one study is by He from China, and there's another study, I forget the author's name, but they have shown that the prevalence of Parkinson's disease is higher in people that have mutations in DMT1. Now, they did not focus on manganese, so I can't tell you whether the levels of manganese in the brains of the folks that have the mutation in DMT1 are actually higher. It's a little bit more complicated because if you think about it, a mutation in DMT1, in order to get more manganese in the brain, because it's an importer, it would actually imply that the mutation is a gain-of-function mutation, which is certainly possible. But they've not looked at that, so they haven't measured any of the manganese. But there is a relationship between some of these transporters. As far as I know, the SLC 3810 is probably the best documented relationship and any disease outcome with a specific transport for manganese. Obviously, we know about Menkes disease and Wilson's disease, other diseases related to other metals, but there is evidence that DMT1 mutations are associated with increased prevalence of Parkinson's disease. Greg, you had a question.
Yeah, so you mentioned it, you didn't show the data today, but tell me a little bit about the MRI study that you did with the neonates that had the parental nutrition. It showed that it showed the elevated levels of the manganese, and were there any differences in development, neurological symptoms, things like that, and one versus those that had normal MRIs?
So you remember Dr. Maitre at the Department of Pediatrics?
Yeah.
She followed those kids for a couple of years. Now, as you know, you're a clinician. The kids that we had in this study that were the controls were not truly controls, because you cannot do an MRI in a child less than 6 years old, even if the parent consent. They must have a reason to undergo an MRI. So these were kids that had, trauma, accidents, or other kinds of things. We compared, I forget how many kids. It was 24, 36 kids, probably 18 in each of these groups. And we looked at various different stages. A lot of them were premature. Most of our controls were not. So there are differences between those two populations. But just in terms of the T1 weighted image, it was actually pretty incredible, because the data, I've never seen anything as clear cut as those MRIs. If I gave you those 36 MRIs, I think you'd be able to place 15 of them very easily into, these are kids that are on parenteral nutrition, and these are kids that are not in parenteral nutrition, and the other 3 in each group you probably would put somewhere in the middle. But it was very clear cut. So they have been looked at 1 year and at 2 years of age. They seem to be slower in terms of speech development. I forget the test where they put, like, it's a encephalogram, EEG. EEG. So they've undergone EEGs. They're different. I can't tell you the details, but they're different. So the firing rates, different regions are different. The problem is that there's so much that, the manganese actually leaches from the PVC tubing. So if you put somebody on parenteral nutrition, there should be enough manganese there without any supplement. And we have written to these companies, and I actually don't know what the status is now, but again, as you know, as a clinician, it's too bad I have the slides here, but I can show you later. I think it has to be more of a precision medicine, to the point where you give the kids exactly what they need. The way those supplements come now, it's a combination of 4 different things. It's selenium, I believe it's copper, iron, and manganese. No, zinc and manganese. It's not iron. They don't give them iron. So there's other things in there. But as a clinician, when you order it from the pharmacy, you can't say, I want X amount of manganese and I want more zinc. It just comes as a solution. And you're pretty much stuck with what's there. It would be nice if, you could do a blood test before and see whether a child is deficient in manganese. If the child is not, if levels of manganese are, 10, 12 micrograms per liter, you wouldn't have to give them any manganese. Yeah, there's.
Yeah, so excellent talk. And I was looking at your EHS data for the SLC, and you have a lot of that data comes from indirect transport as a blood accumulation. You do ever do a direct vascular transport, and you have an idea of the K and Vmax for this transporter and
No, so we have not characterized this. The DMT1 has been characterized very well, all the way from yeast to mammalian system. I think the SLC 3810 is relatively new transporter. So we have not done any Vmax or anything like that. Certainly you could do it, but as far as I know, it hasn't been done. Yeah.
You said that you wrote letters to those companies about the excess of manganese 100 times more than necessary. Have you considered partnering with neonatal nurses and the families that graduate from those units, because they stay connected for multiple years after those neonates graduate? And neonatal nurses are a very unique, very bonded group. They may be a very good advocacy group for you. And there's a history here where phthalates have been removed from tubing in NICUs, so there must be an advocacy way to partner and get that science translation taken care of. Have you thought of?
So, I was not the lead PI on this. I would have to check with Dr. Maitre. She actually moved. She's not at Vanderbilt anymore. I believe she moved to Ohio State. But I can check with her. I mean, this is almost like the most translational studies that we've ever done. I think with the greatest impact on human health. And it would be so easy to rectify the situation. It's the same story. 20, 30 years ago, if you bought baby formula, there was a guy named Bo Lonerdale at the University of California at Davis. He just measured manganese in baby formula that you buy, Similac or whatever. I forget the names of we use them, but it's too long. And there was tons of manganese in there, and the companies actually took it out. Now we have, of course, I don't know if you're familiar, but congressional subcommittee came out with a report last year. They didn't look at manganese, but they found out like 20%, well, not 20%, sometimes 20-fold higher levels of mercury, arsenic, cadmium, and what was another one? Lead in baby food. All the companies, Haynes, the few that didn't have it, and they do analyze, but it seems like the companies, I don't want to say anything, but it seems like these companies knowingly knew what's in there, and they still marketed it. So, I mean, those are issues that, it's all about money for these companies, and human health becomes trivia. It's really unfortunate. I don't know where it's going to go, but, I mean, these are two congressional subcommittees. With lead levels, the other thing that I've learned actually is, like, for example, we have limits for mercury in drinking water, but we don't have limits for mercury in baby food. The FDA has never set up. We do have for lead, we do have for the other metals, but we don't have one for mercury. There is one for drinking water, but not for baby food. I mean, but I agree with you. I mean, somebody should know about it. I can check with Dr. Maitre. Yes.
So you mentioned that one of the limitations of using C. elegans is that they only have 8 dopaminergic neurons and only 302 neurons entirely. When you're looking at diseases like Parkinson's and manganism, where you see specific regions of the brain being degraded, does that pose any limitation to the translatability of the research where you're seeing you can't really look at the degradation of certain structures and the bioaccumulation of these metals?
I fully agree. I'm always the first one to say, I wouldn't extrapolate from, worm epidemiology to humans. So, but I think it's a very valuable tool if you're interested in mechanisms. I showed you dopamine and serotonin, the synthesis, the enzymes, the degradation, the reuptake, everything is exactly analogous to mammalian system. So I think you can actually save a lot of time by deciphering different kinds of mechanisms. I mean, I certainly would want somebody to repeat things in mice and rats, but I think you can do things much more quickly, and it can give you ideas. So I work with Aaron Bowman, for example. A lot of the things that we are doing in C. elegans are informing us on what might be relevant to do in, pluripotent cells. He's working with stem cells. So we ask basically the same questions. He can differentiate his neurons to dopaminergic neurons, serotonergic neurons, then treat with manganese and ask questions You know, are the serotonergic neurons sensitive? And I mean, it becomes closer to mammalian systems. But I think in terms of mechanisms, which always has been my interest, how do things get in? How can you protect? I didn't show you anything about NRF2, but again, it's the same thing. It's instead of NRF2, it has a different name. It's skin, SKN, but it's the same enzyme, same gene.
Thank you.
Yep.
Any other questions?
Online questions or hand raised?
Great, all right.
Yeah, thank you.